Welcome to this video about anti-D. This is part 1 of a 2-part video set. In this video, we will explore the impact of sensitization to the D antigen for pregnant individuals and explain why correct and timely administration of anti-D immunoglobulin is important. There are many blood group antigens carried on our red blood cells. Each one of us has a different antigen set. We're all very familiar with the ABO blood group, but there are other blood groups that are important to consider for blood transfusions and during pregnancy. The D antigen is part of the Rh blood group system. Presence of the antigen is known as D positive and absence as D negative. Our immune system can produce proteins called antibodies when exposed to antigens it does not recognize. These antibodies can attach to the antigen and cause destruction of red cells, which is called hemolysis. If D-negative individuals are exposed to the D antigen, they are highly likely to make antibodies called anti-D. Exposure to the D antigen can occur in 2 ways: by transfusion of D-positive blood components and by pregnancy. When a D-negative individual is carrying a D-positive fetus, transfer of fetal cells across the placenta can occur, leading to development of antibodies by the maternal immune system. The production of anti-D is not reversible. During pregnancy, anti-D can cross the placenta from the mother into the fetal circulation. If the fetus is D-positive, the antibodies can destroy fetal red cells. This is called hemolytic disease of the fetus and newborn, or HDFN. The condition may be mild, resulting in the baby being born with anemia and jaundice, or can be severe with hydrops fetalis, resulting in fetal or neonatal death. The blood product anti-D immunoglobulin, or anti-D Ig, is made from the plasma of blood donors who have made anti-D. In the 1960s, it was discovered that injecting D-negative mothers who had delivered a D-positive baby with anti-D Ig reduced the risk of HDFN in future pregnancies by clearing D-positive red cells before they are recognized by the maternal immune system. Since then, anti-D Ig has also been given antenatally following other potentially sensitizing events, as well as following delivery of a D-positive infant. Anti-D Ig is also given routinely during pregnancy, known as routine antenatal anti-D prophylaxis, or RAADP, in line with NICE guidance. To be effective, anti-D Ig needs to be given within 72 hours of a potentially sensitizing event, including delivery. Normally, a minimum dose of 500 international units is given. Sometimes additional doses of anti-D Ig are required if laboratory testing shows a volume of more than 2 milliliters of fetal cells have entered the maternal circulation, and follow-up testing is then required to ensure all fetal cells have been cleared. When anti-D Ig is required following administration of D-positive blood components to adults or children, the dose will be variable, and advice should be sought from a hematologist. Traditionally, RAADP has been offered to all D-negative individuals, despite approximately 40% of them carrying D-negative fetuses. Non-invasive prenatal screening for RHD using cell-free fetal deoxyribonucleic acid, or CFF DNA analysis, is now available. Fetal DNA is extracted from a maternal blood sample, and testing can be performed as early as 11 weeks gestation. Anti-D Ig can then be targeted to individuals carrying D-positive fetuses, avoiding unnecessary administration of this human blood product. Because anti-D Ig is a blood product, it is important that we have traceability. That is, we know the batch number the individual has received, and also that serious adverse reactions and events are reported. Reactions should be reported to the MHRA via the yellow card scheme, and events to SHOT via the SABRE portal. In the next video, we will look at adverse events and cases of immune anti-D reported to SHOT. More information can be found through the following organisations' websites, as well as through the SHOT website www.shotuk.org noninvasive.org/resources/current-resources.